The human brain is wired to experience sexual fantasies, a phenomenon rooted in the limbic system. Studies show 95% of adults fantasize, with dopamine playing a key role in arousal. fMRI scans reveal heightened activity in the amygdala during fantasies, linking emotion to imagination. This mental rehearsal may enhance real-life intimacy and stress relief. When encountering an attractive stranger, the brain releases phenylethylamine PEA, creating euphoria. Oxytocin levels rise by 40% during eye contact, fostering trust. Research indicates this chemical cocktail mirrors the effects of cocaine, explaining intense initial attraction. The ventral tegmental area becomes hyperactive, triggering obsessive thoughts about the object of desire. Neuroscience proves anticipation activates pleasure centers more than the act itself. A Princeton study found the nucleus accumbens shows 300% greater activity during anticipation versus consummation. This explains why delayed gratification and teasing touches heighten arousal. The brain's reward system values the maybe more than the certainty in sexual contexts. Power play activates distinct neural circuits. Dominance behaviors stimulate testosterone production, while submission triggers endorphin release. Yale research reveals 68% of fantasies involve power exchange, linked to childhood attachment patterns. The prefrontal cortex temporarily disengages during roleplay, allowing for immersive experience without judgment. The brain integrates multisensory input during arousal. Touch activates the somatosensory cortex 50% faster when combined with whispered words. Scents bypass the thalamus, going directly to emotional centers. This explains why certain perfumes can trigger vivid memories. Princeton studies show combining 3+ senses increases pleasure perception by 75%. Mirror neurons fire both when performing and observing intimate acts, creating empathic pleasure. UCLA research shows these neurons are 40% more active during mutual gaze than during sex. This neural mirroring explains why seeing one's desire reflected intensifies the experience. It's the biological basis of chemistry between partners. During climax, the brain shows patterns similar to epileptic seizures. The hypothalamus releases oxytocin in pulses, while the amygdala temporarily shuts down, explaining loss of fear and inhibition. PET scans reveal the prefrontal cortex deactivates, creating timelessness. This neural fireworks show is the most complex response the brain can generate. After climax, the brain floods with prolactin, causing relaxation, while serotonin rebounds create emotional bonding. This 30-minute refractory period shows unique delta wave patterns. Harvard studies found cuddling maintains elevated oxytocin for hours, strengthening attachment. The ventral pallidum becomes active, associating the partner with reward. Brain scans show fantasies activate the same regions as real experiences, but with 30% less sensory cortex engagement. This explains why imagined scenarios feel vivid yet safe. The temporal lobe stores these fantasies as pseudo-memories, blurring lines between real and imagined encounters over time. Anthropologists suggest sexual fantasies served evolutionary purposes, practicing mating strategies risk-free. Modern neuroscience confirms they maintain libido during partner separation and explore desires safely. The brain's ability to derive pleasure from imagination alone is a unique human adaptation, with 85% of women and 72% of men reporting fantasy-enhanced arousal.